Hello. Today I'm going to be talking about RNA interference, or RNAi. First off, I'm just going to take a step back and talk about the central dogma of biology because this will come into play with the whole RNA interference. So the central dogma of biology is that DNA codes for RNA, which then codes for protein. And in RNA interference, what I'm going to be talking about, specific types of RNA, they're called either small interfering RNA or microRNA, inhibit this process of translation of the mRNA into protein. So that's what's going to be talked about going forward. Okay, so now I'm going to talk briefly about just the general biological concepts sequence that is going on here before going into this nitty-gritty chemistry explaining exactly how it occurs. So the first part of this whole process is the introduction of double-stranded RNA into the cell. And now this double-stranded RNA, as you probably know, RNA is single-stranded usually. And so this double-stranded RNA is unique, and it's specifically made, like genetically engineered, to be complementary to a sequence of nucleotides that make up a gene that we're looking to target and influence and stop and shut off because we don't want this gene to be transcribed. So this double-stranded RNA is introduced to the cell, and this acts as a signal to attract dicer, an enzyme that cuts it into small pieces and cuts it into single strand as well. And these pieces are called RNAi, or like small interfering RNA, or microRNA. And so these RNAi fragments then also combined with argonaute protein to form the RNA-induced silencing complex. And this complex acts kind of as like the scissors, the machine. Now with the specific sequence on the siRNAs that we've coded for when we made the double-stranded RNA to be introduced into the cell, that specific sequence is now going to be able to search the mRNA, which is like all the coded transcribed DNA. And once the right gene, this desired gene, is found, it'll then bond due to complementary base pairing, which I'll go into a bit later. And then the gene can be cut out of the mRNA and destroyed. So basically, just being really short, there's double-stranded RNA. It gets cut. It gets cleaved into smaller little pieces that are specific to the gene that we're looking to bind to and silence, which then forming the RISC, or the RNA-induced silencing complex, then acts as this mechanism to cut the gene. So now I kind of wanted to talk about the specific chemistry of why this process can occur. So the most important concept with this whole process of RNA interference is the specificity of how the microRNA can match up perfectly with the mRNA sequence that corresponds to the gene. And the way this is done is due to the specificity of the base pairs, and specifically hydrogen bonding. So as we've learned in class, hydrogen bonding is a specific type of a really strong intermolecular force in that when one really electronegative atom, such as oxygen or fluorine or nitrogen, is bonded to a hydrogen. And so when this causes a strong polarity shift of the bond, which kind of exposes the proton on the hydrogen, and this exposed proton is very strongly attracted to the other electronegative atom of the pair. So if you look at this slide here on the nucleotide interaction, you can see that the guanine and cytosine match up perfectly with the hydrogen bonding. And if you were to imagine, say, if you were to look at adenine and thymine, if you looked at adenine and imagined lining up cytosine, the hydrogen bonding would not line up, and so the bond cannot be formed, and so therefore the wrong pair cannot be made. And so what this means is it means that the specific nucleotide sequence we put in the double-stranded RNA in the beginning, engineer it, scientists can make sure it'll get matched up with the exact right sequence of gene that we're looking to correspond to cut up. Okay, so now I wanted to talk about how the RISC complex actually breaks the mRNA backbone when it's binded and it's located the gene it wants to cut. And so the specific bond that's actually cut is called a phosphodiesterate bond. And this is between a phosphate group and the adjacent sugar, ribose sugar in the mRNA. And so, in order to break this bond, we actually have to go through a process called hydrolysis, or the addition of water to lyse the bond. And so, this process is actually not like exothermic by itself. So what RISC does as a giant protein with all these different activation sites, it actually contains regions in its molecular shape that allows for the splitting of the water into the hydrogen ion and the hydroxide ion, and these ions work together to cleave the nucleic acid by reforming the hydroxyl groups on each nucleotide, as you can see on the left here on this diagram. And although the splitting of water is certainly endothermic, the activation energy of this reaction is lowered via the catalytic properties of RISC. And so also it's worth noting that like this overall reaction is exergonic. It has a delta G value of negative 5.3 kilocalories per mole. Part of this has to do with the fact that when you split it into two nucleotides, this increases the entropy of the universe. And also by separating the negative charges, you actually lower the electrical potential energy of the system. Lowered activation energy, the increased entropy of the universe, and the formation of more energetically favorable bonds at the end of the process overall together make this process energetically favorable. The significance of RNA interference is that scientists have discovered certain genes that are responsible for the conditions like Huntington's, Alzheimer's, and cancer with its BRCA genes and P52 genes. So if you can imagine, this is kind of like an application of genetic engineering. If you know the specific nucleotide sequence for a gene that we want to silence that might help prevent a certain cancer or a certain condition, you can genetically modify double-stranded RNA to have the complementary base pair sequence, package this into some sort of virus or some other vector, inject it into the body and tissues, which will then release the RNA fragments into the gene, into the cells that we want to target, and then that can go and undergo the same process I just explained of how it can silence the genes that are desired. So now that I've kind of mentioned the potential for application, I kind of want to talk about exactly what progress is being made to turn this mechanism into a functional drug or a functional pathway for medicine. So there's one company called Analam, and it's actually performing siRNA therapy on rats at the moment. And it's actually shown great success at targeting the ALS and Parkinson's genes in the central nervous system of these rats. And these rats actually have models for its central nervous system that are very similar to that of humans. So it's actually a very good discovery to have experienced. And because of that, they're kind of expecting an experimental drug to be available in early 2020. But the real problem is, if you imagine the double-stranded sRNA is the key here. This is the thing that we can engineer, that we can make target the specific gene we want. But it's unstable. You know, RNA is usually single-stranded. So what needs to be done in the future to make this drug a reality is there needs to be a better vector to transport the double-stranded mRNA, double-stranded RNA, so that it can reach the tissues that are going to be targeted and be uptaken by the cell. So right now, the uptake by the cell is also a bit of an issue because it is this foreign object that's not really produced in the cell organically. So the future is to find a way to stabilize this molecule, stabilize and direct the vector in a more precise manner, and increase the uptake into the cell. Overall, if they can make these changes and make it more stable and less degraded, really a lot of promise for this method in the future. I hope you learned something. I hope you enjoyed my video. I kind of just gave my presentation and showed you my slides, but this is more or less what I would have given in class. And I just wanted to say on one final note, thank you, Professor Sung, so much for all that you've taught us. I really enjoyed the class, and I hope you have a great holiday break.